Hi, welcome to my YouTube channel Bio Explorer. I am the Mujjaratna. This is an educational channel and it will be very useful to improve your biological knowledge. Today in this video we are going to discuss about nucleic acids. Nucleic acids. They are another class of main organic compound found in organisms. Hereditary information and the information needed to make proteins when necessary are stored in nucleic acids, mainly in DNA. Nucleic acids are polymers exist as polynucleotides made up of monomers called nucleotides. Main elemental composition is carbon, hydrogen, oxygen, nitrogen and phosphorus. Nucleic acids are macromolecules and biopolymers. There are two types of nucleic acids as DNA and RNA. The structure of nucleotides. They are the monomers which make nucleic acids. Millions of nucleotides combine to form a linear chain called a polynucleotide. Each nucleotide is made up of three parts as pentose sugar, nitrogenous base and phosphate group. A nucleotide without a phosphate group is called a nucleoside. Example, adenosine and guanosine. Pentose sugar. The sugar molecule has a central position in the nucleotide. Pentose sugars are of two types as deoxyribose and ribose. Deoxyribose is the sugar in DNA and RNA contains ribose. These two sugars look similar in structure. The only difference is that there is a hydroxyl group connected to second carbon in ribose, but there is a hydrogen atom attached to the equivalent carbon of deoxyribose. Nitrogenous base attaches to the first carbon while phosphate group attaches to fifth carbon. Nitrogenous bases. They are carbon-based molecules which consist of nitrogen-containing ring structures. There are two major groups of nitrogenous bases as purines and pyrimidines. Purines are nitrogenous bases containing two fused rings. In purines, there are two types, namely adenine and guanine. Pyrimidines have a single carbon-nitrogen ring, and there are three types of pyrimidines as thymine, uracil and cytosine. Bases are commonly represented by letters as A for adenine, G for guanine, T for thymine, U for uracil and C for cytosine. Each base has a unique structure with various functional groups attached to it. Nucleotides of DNA contain adenine, guanine, thymine and cytosine. In RNA nucleotides, they also have adenine, guanine and cytosine, and they have uracil instead of thymine. Phosphate group. It gives the nucleic acids the acidic nature. Phosphate group is attached to the fifth carbon of the pentose sugar. Nucleotides may contain a single phosphate group or a chain of three phosphate groups. In a chain of DNA or RNA, each nucleotide bears a single phosphate group. Formation of nucleic acids. Nucleic acids are composed of linear chains made up by millions of nucleotides. Those nucleotides are connected by phosphodiester bonds. Two neighboring nucleotides undergo a condensation reaction resulting in a phosphodiester linkage. This bond is formed between the hydroxyl group of the phosphate of one nucleotide with the hydroxyl group attached to third carbon of pentose sugar of the other. These bonds result in a backbone with a repeating pattern of sugar phosphate units. This sequence of nucleotides has a directionality. because two ends of the polynucleotide chain are different from each other. Those two ends are called 3' end and 5' end. At the 3' end, the hydroxyl group attached to third carbon in the pentose sugar of the last nucleotide is exposed. At the 5' end, the phosphate group attached to fifth carbon of the first nucleotide is exposed. The structure of DNA molecule. DNA consists of deoxyribonucleotides. James Watson and Francis Crick, based on the X-ray crystallography of a DNA molecule, proposed the double helix model to describe the structure of DNA. According to this model, DNA molecules have two antiparallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. The sugars and phosphate groups that lie on the outside of the helix form the sugar-phosphate backbone. The two sugar-phosphate backbones run in opposite directions from each other. That is, when one backbone runs from 5' to 3' end, other goes from 3' to 5' end. This arrangement is referred to as antiparallel. Nitrogenous bases are paired in the interior of the helix. The bases of a pair are bound to each other by hydrogen bonds. The two strands are held together by those hydrogen bonds. The structure of DNA is also similar to a twisted ladder. The rails of it are composed of sugar-phosphate backbone, while the steps of the ladder are paired nitrogenous bases. Base pair rule. Any two bases can't be paired in the double helix. The nitrogenous bases are different in size and have different functional groups. So base pairing is highly specific and always a purine base pairs with a specific pyrimidine base. Adenine can bond with thymine by two hydrogen bonds in a DNA molecule, and guanine can only pair with cytosine by three hydrogen bonds. Hence, two chains are said to be complementary to each other. Those pairs are known as complementary base pairs. In this original double helical structure, one complete turn consists of 10 base pairs. Functions of DNA. Store and transmit genetic information from one generation to the next generation. In most organisms, DNA is found as the genetic material. However, some viruses consist RNA as genetic material. DNA is found in the nucleus of eukaryotic cells and also found in specialized cell region called nucleoid in prokaryotic cells. A particular site of a DNA chain is a gene. DNA and associated proteins form structures called chromosomes. A chromosome may contain tens of thousands of genes. Those genes provide information to perform various functions and to produce different products in the cell. Store genetic information for protein synthesis. Many genes encode protein products. The information for protein synthesis are stored by the sequence of base pairs in a gene. This sequence is copied to make an RNA molecule. Then it is decoded to specify the amino acid sequence of a polypeptide. The structure of RNA. This is normally a single-stranded nucleic acid. It is composed of ribonucleotides containing uracil, cytosine, guanine, and adenine as nitrogenous bases. We can identify complementary base pairing between two RNA molecules, so within the same molecule. Complementary base pairing facilitates three-dimensional shape that is essential for their functioning. There are four major types of RNAs as messenger RNA, transfer RNA, ribosomal RNA, and regulatory RNA. Messenger RNA. It is a linear molecule. When the cell needs to synthesize a particular protein, a RNA copy of the relevant DNA sequence will be made. It copies only one strand of DNA. This carries the same information as the gene, but because it is an RNA molecule, thymine in the DNA is replaced with uracil in mRNA. Then, this RNA copy will associate with the ribosome to perform next steps to produce the protein. mRNA is the least abundant type of RNA in a cell comparatively. We can mention the functions of mRNA as copies the genetic information stored in DNA molecule as a sequence of nitrogenous bases, transports genetic information from nucleoplasm to the site of protein synthesis through nucleopores. Transfer RNA. It is the smallest RNA molecule. It is considered as linear because it consists of a single strand of RNA, but complementary segments of this strand bound together to make double-stranded regions. This forms three-lobed structure important to the function of the molecule. The main function of tRNA is the transportation of amino acids to the ribosome, which is the site of protein synthesis. Ribosomal RNA. It is the most abundant type of RNA. It is also a major component of ribosomes. rRNAs have a complex irregular structure. It helps mRNA to bind in the right spot in the ribosome and provide the site where polypeptide chains are assembled. Regulatory RNAs. They do not encode proteins. They help to regulate the expression of other genes. Examples: microRNAs and small interfering RNAs. So that's for today. Hope you enjoyed this video and gain a lot of interesting facts about nucleic acids for your biological knowledge. Please subscribe this channel and click the bell icon. See you in the next video.